Well, thank you, Glenn. I apologize for being late. I had great problems logging into the system this morning. But I'd like to thank the organizers for their very kind invitation to present this talk on the history of the International Symposia on urolithiasis. The first meeting took place in Leeds in 1968 when I was finishing my PhD studies at that time, and then moved around to 14 different sites around the world, roughly every 4 years. And we covered all 5 continents, finally finishing up here in Athens. This is the proceedings of the very first meeting in Leeds, which was run by my boss, Christopher Nordine. And I've set it here against my collection of kidney stones, which I collected around that same time period in the mid to late 1960s. And the difference is that the stones then all came out as whole stones, because the only operations available to surgeons at that time were open surgery and perhaps Dormier basket. So you can see the wide variety of stones, which you never see nowadays, because all we get from the urologists are little piles of dust. The meetings proceeded around the world every 4 years. And these are the proceedings from the meetings during that time up till 2004. After that time, it became too expensive for conference organizers to print proceedings, and so there is none after 2004. In that very first meeting in Leeds, when as I say, I was a PhD student at that time, I presented the very first paper on supersaturation in urine, and that was taken from data in my thesis, which was on the physical-chemical aspects of renal stone formation and published at that time, a year later. In my thesis, I made a note in the introduction that it seemed to me that throughout the history of research into stone formation, many theories have come and gone during that time, but like an ever-expanding spiral, which at each turn almost returns to its original position, apparently obsolete theories reappeared decades later. And as the spiral slowly winds round, more is added to our general understanding of the factors involved in stone formation, but a single cause, if indeed such exists, has yet to be uncovered. So that's what I wrote in my thesis in 1969. And here's what I mean. Here is a spiral showing increase in knowledge over a period of time. But do we ever get to the answer of the cause and treatment of stones? If we go back to the origins of modern research in stones, we start, say, with Randall's plaques in 1937. Then they reappear again in 2003 with the work of Fred Coe and his group. If we look at work on the influence of cells and renal tissue on stones, we have the lymphatics theory of Carr and Bradford and the Hamilton-Stewart's Nest. And that has come back, of course, later in the crystal cell interaction work of Said Khan and, of course, many others around the same time. Supersaturation was first sort of suggested as a main factor in stones by Vermeulen in '59. Then I wrote the very first supersaturation program, as I say in my thesis, in 1969, and that was followed 16 years later by the publication of the famous ECHO program by Vernes et al. Then there's the matrix theories, the importance of matrix in stimulating stone formation, attributed mainly to Boyce in 1959, but recurs later in the work of Marty Resnick. Then there's hypercalciuria, which really goes back to Hodgkinson's work in the early 60s, but then reappears in the work of Pack and Co. in the 70s. Then comes the inhibitor period, when it started by Herbert Fleisch, followed up by John Eager Howard a few years later, and followed later than that by the lifetime work of Rosemary Ryle on various inhibitors of crystallization. And then more recently, we come to the epidemiological factors, metabolic factors, genetic, nutritional, and lifestyle factors, and almost everybody has contributed to that. But the very famous bone scientist in America called Bill Newman, and I remember him at a conference, I wasn't at the conference, but I remember it from the proceedings. He said, if you ever think you've found something new in science, go back and look in the old German literature because you'll find it there. And sure enough, when I went back, I was just about to publish my thesis when I found the very first elements of a supersaturation program written in a German paper by Meyer in 1928. And if you look at the modern aspects of epidemiology, you can actually go back to around 400 BC with the work of Hippocrates. In his earliest publications by Hippocrates, he pinpoints a number of factors which we still study today. The importance of seasons of the year, temperature, as we've just heard from Alberto, quality of water, lifestyle activities and the mode in which inhabitants live, diet in terms of overeating and drinking to excess, or whether you have a life of exercise or perhaps a much lazier life. And it was Hippocrates who first advised stone formers to drink more fluids to stop them forming stones. He called it wine mixed with abundant water, which sounds a good solution to me. And he also discouraged his pupils from cutting for stone. So from Hippocrates in Kos around 400 BCE to Athens this year, have we learned anything new about stones during the 54 years of the ISU Symposia? I'm sure we have, but research seems to have been going round in circles to get there. And we've ended up even going back full circle to the teaching of Hippocrates 2,400 years ago, where research on stones actually started. As my boss in Leeds once said, that is why they call it research. Research means look again. In the meantime, during these 54 years, urologists have made great advances in the urological management of stones. When I first came into the stone field in the early 1960s, only open surgery or dormia basket were the methods for removing stones available to surgeons. Then in came rigid ureteroscopy, PCNL, SWL, now up to flexible ureteroscopy and EPVL. And what next do the urologists have up their sleeves for removing stones? And this is represented actually in the pattern of presentations at ISU Symposia. What I've plotted here are the percentage of presentations. I've gone through all the proceedings and programs of the meetings during that time, and I've divided them into what I call research presentations and those based on urological management of stones. In the first 4 years, 100% of the papers presented at these meetings were on research. Then, as SWL came in, gradually more papers appeared in the proceedings on the urological management of stones, and that remained constant for a number of years until the advent of the modern PCNL and flexible ureteroscopy methods, when the percentage of papers presented at these meetings has really overtaken. And in fact, this year there are more presentations on urological management of stones than there are on the research into the causes of stones. Now, of course, partly that drop in the research is due to difficulty in getting funding for research. So what is the future for non-neurological stone research, if any? Well, I've identified 5 possible ways from the literature that we could go in the future. First of all, there is an increased evidence of studies on the genetic defects that may lead to stone formation. At the recent meeting in Brussels, they're talking to the nephrologists and geneticists there. It's currently estimated that 60% of stone patients have some underlying genetic defect that increases the urinary excretion of one or other of the various stone-forming ions, particularly calcium. And here are a list of the genes so far associated with stone formation. Apparently there are 48 that have been identified. I found 43 on this list. Many of them, as I say, are involved in calcium, increasing urinary calcium, but others are involved in urate stones and, of course, cystine stones and so on. So, and this slide is from courtesy of John Sayer. And if you're interested in looking at the genetic aspects of stone disease in more detail, I'll refer you to a special issue of the journal Urolithiasis in February 2019, which was guest edited by Giovanni Gambaro on the genetic aspects of stone disease. And you'll find a lot of articles there that will bring you up to date on that topic. Moving on to enzymatic treatments designed to destroy oxalate in the intestine. There are 2 enzymes, as you know, which can degrade oxalate. One is called oxalate decarboxylase and the other oxalate oxidase. And the oxalate decarboxylate has been used in a study here using a recombinant microbial oxalate decarboxylase ALLN177. And this is a study by Craig Langman on that. And what he did was they studied a group of patients on a basal diet. They then put them on a high oxalate, low calcium diet, and of course urinary oxalate increased markedly on that. And while they were still on that diet, the patients were divided into 2 groups. One were given placebo and the other half were given this drug, the oxalate decarboxylase containing drug. And although the effects are actually significant, but not markedly so, but it did reduce urinary oxalate on average by about 11 milligrams per day. So perhaps there is some way that that sort of technology can be used to reduce oxalate in future. More recently, there have been papers using RNA interference therapy, in this case, again, to reduce endogenous oxalate. And this is a study by David Sass at the Mayo Clinic using a new drug called lumasiran, which has been given to children with primary hyperoxaluria. And this shows the data on the reduction in urinary oxalate in the 8 patients studied so far, month by month on treatment. So that looked very promising way of perhaps reducing urinary oxalate in future. So I think that is a technique that we need to keep an eye on. Then there are possibility of chemical treatments designed to fragment stones in situ. And one example of this was published by John Aplin about 6 years ago, using a drug called hydroxycitrate. And here is its structure compared with that of citrate. Hydroxycitrate has an extra hydroxyl group there, as you can see, which imparts quite different properties to the hydroxycitrate compared with the citrate itself. First of all, it's absorbed more strongly because of that extra hydroxyl group onto calcium oxalate crystals than is citrate. It's a more powerful inhibitor of crystal growth than is citrate. It imparts artificial strain on the crystal lattice of the calcium oxalate crystals in stones, and this leads to calcium and oxalate being released into the solution to alleviate that strain, and eventually leads to the dissolution of calcium oxalate crystals. And very importantly, this takes place even in supersaturated solutions of calcium oxalate. So that's a very new phenomenon. And if that is true, that is a potentially a fantastic way of perhaps breaking up stones chemically. It can be given orally, and it is excreted in urine. There are some suggestions that there may be some side effects from this drug, so this may not be the one that's finally used, but the concept is an important one, I think. And then finally, there is the possibility of the use of nanotechnology to prevent new stone formation or even to fragment existing stones. Nanoparticles, which are particles smaller than 1,000 nanometers, that's 1 micron, and these have a very high surface area to volume ratio, and it gives them quite different properties to those of materials with dimensions at the micro and macro scales. And I have to thank Rosemary Ryle for this slide. So possible applications of that type of therapy would be to design and synthesize molecules based on peptides or proteins that can absorb strongly to stone mineral crystal surfaces. In fact, manufacture of nanoparticles similar to those based on hydroxyapatite are already in use for targeted controlled release of drugs. Prevents crystal formation or enlargement or attachment, or even to enhance intracellular disposable of crystals, or possible for the possible use as for transfecting oxalate oxidase or oxalate decarboxylase into the genomes of recurrent patients with primary hyperoxaluria. So the final message is, as I would see it, the urological removal of stones is not the ultimate answer to the management of stones patients, since it does not cure the underlying cause of the problem. So I would encourage you to get involved in multidisciplinary research into finding ways of preventing stones from being initiated and or to stop the initial crystals from developing into stones. So I would suggest that you leave your sophisticated lithotripters and flexible ureteroscopes for part of the day and get into the laboratory with your scientists. But that way, the true solution to the problem may be found. And I leave you with a poem by the 16th and 17th century English poet called Francis Quarles. It's called Respici Finem, which means look to the end, wait till you get to the end. My soul, sit thou a patient looker on, judge not the play before the play is done. Our plot has many changes, Every day speaks a new scene. The last act crowns the play. Or in more modern vernacular, it's not all over till the fat lady sings. But from a somewhat overweight man now, thank you for your attention. Thank you.